Bradford Patton. I'm a graduate student at Meharry Medical College studying data science. My project is a comparative study of knowledge graph embedding models for drug repurposing of rare diseases. For some background and objectives, there are about 7,000 rare diseases that impact 300 to 400 million people worldwide, with 30 million in the United States alone. Discovering these treatments for these diseases relies on understanding their molecular basis, but this can be a labor-intensive and costly task. However, today's machine learning techniques make it easier to understand these diseases. But with these techniques, however, they face limitations in integrating different data sources and providing explanations for this data. So this is where knowledge graphs come into play. Knowledge graphs they help interconnect heterogeneous biological data, which provides a more comprehensive analysis for your data. And along with that, knowledge graph embedding helps represent this data in a low-dimensional space, enabling the creation of a predictive models for link prediction and node prediction. So with this, in this project, we present a comparative study of knowledge graph embedding models on a large biomedical knowledge graph. We then apply the best performing models in a case study for mesothelioma. This is a rare type of cancer that generally affects older people. This study serves as a benchmark for future experiments with other rare diseases and other knowledge graphs. So for our data, we encompass various biological data sources. At first, we reproduced a bio-KG knowledge graph, which had plenty of biological data sources involved with it. These data sources had relations. We had including drug-drug interactions, drug-disease associations, protein-disease associations, etc. And with this bio-KG already reproduced, we combined some rare disease data from Pharaoh's database to combine with the bio-KG data. We took the mesh IDs and the uniproc IDs. And after we combined it, we made it into one called rare disease database. So with this rare disease data, to actually train the data, we split the data into 80-10-10 split, where we would have a training, validation, and testing data set. So in this drug repurposing pipeline, first we perform our data integration, where we take the bio-KG data and we take the Pharaoh's rare disease data and we implement that together. Then after, we create a knowledge graph, rare disease knowledge graph. Then after, we could train our models. These models could be semantic matching, translational distance, or neural network model. And then we evaluate our models off these metrics: mean rank, mean reciprocal rank, and mean and hits at K. This could be a hits at 1, hits at 5, or hits at 10. After running these models, we get our embeddings. Then we can perform our link prediction task with our models and get the repurposed drugs. So after running these models, we wanted to specifically look at the hits at 10 to see how far down the list the correct drug appeared. So our threshold started at 0.5. If it was below 0.5, we didn't count that as a good performing model. If hits at 10 was above 0.5, we count that in a good performing model. And for the performances, we had about 6 models that were above 0.5. So with the outcome here, we can get some drugs, cancer-related drugs that are involved with the data in our graph. So we have Cross-E, Mir, Proggy, Complex, and Quake. So with this, we get the current uses of these drugs. And these are all either cancer-related drugs or investigational drugs. And these drugs were ranked in each model. So we may have a drug coming in first place, or a drug coming in second in the list, or a drug coming in fifth in the list. These were the best performed models, which we ranked these drugs to predict for mescaline. So I would like to acknowledge the NSF HBCU EIR grant, and I would like to thank Dr. Sarker for the travel support from the School of Applied Computational Sciences. Thank you, and I hope you enjoyed my talk.